Welcome back to the SS Enterprise. So now we're going to be talking about more of the details on DNA structure. So let's start with that. I'm going to skip the recap because I just made a recap video. So go ahead and feel free to rewatch that if you need more help with this. So at the moment, let's continue. DNA structure. Remember, we have our two strands and they are connected to each other. The way that DNA works is that each of these strands is the exact opposite, right? So they match up to their opposite parts. You can see here that the shapes match up, right? The C and the G are a perfect fit for each other. The A and the T are a perfect fit for each other. And so they're bonded with each other. And the backbones that hold all of these nitrogenous bases are the sugar and phosphates that are connected there, right? So the nitrogen bases, we said last time, are the ones that are going to tell us the instructions, right? They're the ones that actually matter. These sugars and phosphates are important too. They're the ones that hold this whole structure together. But the ones giving any sort of information are the nitrogenous bases, okay? Now, we do have it twisted around in a double helix, but if you were to unfold it and just stretch it out, this is kind of what it would look like, okay? So, in our twisted ladder model, what we have is also beginners and enders, right? So at the beginning of a strand, we tend to have a 5' is what we call it, right? And then it ends on the 3' end. Now, the opposite side, of course, everything's going to be backwards. The other side, we would have the 3' at the top and the 5' at the bottom, because again, we're just turning it over. Because now the A needs to match with the T, the C needs to match with the G with the C, the T with the A, right? So we just grabbed the exact same information and turned it around. It got flip-flopped. So this is what's going to allow these pairings to happen. Remember, they only fit with a specific pair. You can't mix match these. So these nitrogenous bases, what we knew is that they are held together by hydrogen bonds. However, recently it's been found out that that's not 100% accurate. There is a similar bond to the hydrogen bond, but it's not technically a hydrogen bond. However, in STAR questions, they'll still ask you about hydrogen bonds. So just know those are the ones holding the nucleotides together, okay? Hopefully by next year, we'll be able to like update the textbooks and the people that write these tests, and they'll know that it's not really a hydrogen bond. If you're more curious about that, you can check it out. There will be some links to some articles in the description down below if you're curious about that. So yeah, I just thought it was interesting for y'all to know that it's not 100% accurate, but we still have to teach it that way, sadly. It takes a long time for science advancements to be taken seriously in schools and like that. It's hard for that process of changing everything when we find something we thought was true is no longer true. So that's kind of neat. So the steps here, or the center of our DNA ladder, are these what we call base pairs. So the pairing of the two nucleotides and the bond holding them together. That is what the genetic code is, right? So this section is the information, the genetic information. Now, every living organism is made up of the exact same 4 nucleotides, right? Of the exact same 4 nitrogenous bases in our nucleotides. Now, we don't all look the same, right? Octopus and a duck and a person don't look the same. Bacteria also have DNA and they don't look the same as us, right? Trees and monkeys don't look the same. Ladybugs and mushrooms don't look the same. So how can we all be made up of the same stuff if we look so different, right? Well, the order that we put these letters in makes a difference. So somebody could have this first reading in their ladder, while somebody else may have this in their ladder, right? And so the order in which you put those A's and C's and G's and T's, that is what makes us different. And I know it feels like there could only be so many combinations. How are we so different? Well, like I said, that strand could be about 2 meters tall. And when it's all crumpled up together, it fits inside of the nucleus of a cell. So cells are super tiny. We can't see them, right? So imagine how tinier the DNA is. And so if it's that tiny, when you stretch it out, if it becomes 2 meters, each of those 2 meters are going to have like a whole bunch of information in them. So yeah, there's a whole lot of letters that you're going to have in just one DNA sequence. And so if you just change one letter, it can make a complete difference. And we're going to talk about in class just some examples of some animals and some organisms that are literally just a few letters different from each other. Now, it's not just that you have the same amount of each one, but the order matters. So A, is not the same thing as C, A, right? Order is extremely important. And now we're going to talk about the rules of how we match up these A's and C's and G's and T's. So we have two kinds of nitrogenous bases. We have the purines and the pyrimidines. The purines match with a specific pyrimidine. They can't fit with anything else. They can't fit with each other. And so a purine must bind to a pyrimidine, but it has one specific one that it has to bind to. For adenine, that's thymine. For cytosine, that's guanine. And so these two are always going to be together. A and T are always going to be together. Cytosine and guanine, C and G, are always going to be together. So if you have a C on one side, you know you're going to have a G on the other. That's the only thing you can have on the other side, right? And if you have a T, the only thing you can have on the other side is an A. So that makes things easy, right? So to remember which ones are which, purines, you can remember the phrase pure as gold. Purine, A for adenine, and G from gold for guanine. So pure as gold. And then for pyrimidines, you can remember cut the pie. Cut from cytosine, the from thymine, pi from pyrimidine. Okay? Hope that helps. Now, holding the two strands together, we talked about these hydrogen bonds. Again, let's just keep saying they are hydrogen bonds for the purpose of this class, because we don't really have to go into the details of what holds the DNA together in here. So remembering, again, to match up each other, adenine and thymine, so you can think of angry tigers, okay? And then cytosine and guanine, calm giraffes. If that helps you, use it. If it does, great. If it doesn't, and you can come up with a different one and you want to share it with us, just send it to me and I'll share it with the class. Maybe that'll help them even more, right? Now, we call this complementary base pairing because if you think of it like a puzzle, these are the only two pieces that fit together, and these are the only two pieces that fit together. So we're going to do a lot of practice on this, but like I said, the order of these matters. So if we have one strand rating as this one is, when we ask for the complementary strand, what we're asking is what would match it with it on the other side, right? So if we start with an A, it's going to match with a T. T is going to match with an A. T, A, G, C, C, G, C, G, A, T, and so on, right? So you're just, if I ask you for a complementary strand, I'm just asking which one would match with each letter in the correct order. Okay, so if I start with an A, I have to start the complementary with a T. Everything needs to remain in order. Okay, now for RNA, this is slightly different. For RNA, you still have adenine and guanine as the purines, but now one thing changes. Remember, we don't have thymine in RNA, so our pyrimidines here are cytosine and uracil. So just like thymine paired up with adenine, now uracil is going to pair up with adenine. Cytosine and guanine are still together. So we still have our calm giraffes. So again, purines, pyrimidines. And that's it. That's all of DNA and RNA for today. I just needed you to know the structure, how we assemble them. And remember, since DNA is a double-stranded, that's why we have that complementary strand. RNA is just one strand, so we don't need to have a complementary strand just yet. So make sure you study this up. If you need to go back or slow me down or whatever you need to do, you know, that gear icon is there for you to, you know, change the speed, or you can turn on captions if those are available. And if you have any questions, don't be afraid to reach out to me and send me an email or show up to office hours. But we will be talking about this in class tomorrow, so make sure that you've watched this video before you go into class to make sure you have some of this information still fresh in your mind and we're able to move through it a little faster, okay? So I'll see you in the next video. Bye-bye.